these domains. Is the mic working? I can't tell from it. Yeah, okay. So again, in the previous lecture, we sort of set the stage for the three domains, talked a little bit about the tree of life itself and where these three domains come from. And we're going to talk today about using the rooted tree of life to track the biology of these organisms. And then in the next lecture, we're going to go into, in more detail, the diversity within two of these groups, the bacteria and the archaea. And so we'll start with this tracing the features, and then we'll talk a little bit about the diversity within the domains of life. So I started in on this in the last lecture. We're going to take the rooted tree of life as a given, and we're going to overlay features onto this tree and use the character state reconstruction methods to infer the history of the features. And the main reason to do this is, are you getting a little feedback from the mic? Or I can just move it a little further away. That might. Is that better? No. Yeah, the batteries have been a problem. It died during the last one, and I don't know if they got new batteries yet. But is that better? A little bit? I'll try to be quiet and calm up here. So, many people are interested in the actual history here, but that's not what we're really talking about. What we're talking about here is the biology of these organisms. And we use the history to help us understand their biology and to help us look for features that are important or interesting or worth studying in more detail. So I started in on this one example, and we'll go through a couple of examples. One feature we can track the evolution of, and that is interesting biologically, is the composition of the cell wall in bacteria. The cell wall in bacteria is made up of this molecule called peptidoglycan. And there are a couple of bacterial taxa that don't have a cell wall, and they've thrown away this peptidoglycan, but it's generally found across the entire domain of bacteria. And it's not present in eukaryotes or in archaea. Archaea have cell walls too, as do some eukaryotes, but chemically they're different. So if we take this rooted tree of life, we can treat peptidoglycan as a character, and the presence of peptidoglycan as a character state, and the absence as a character state. And if we, in this case, color the presence in red and overlay that onto the tree and the absence in blue, we're going to ask, what are the possible scenarios for character states in the ancestral nodes here in the tree, and reconstruct the possible history of character states. So here's one possible scenario. The most recent common ancestor of the three domains of life had peptidoglycan, and then it was lost in the lineage leading up to the common ancestor of archaea and eukaryotes. A second possibility is that it was absent from the most recent common ancestor and gained in the lineage leading up to the common ancestor of all bacteria. If we compare these two hypotheses to each other, they both require one character state change. So given this information and this information alone, we can't distinguish which of these two patterns is the best pattern. That makes sense what we're doing here? We're just reconstructing, treating these as the same type of character states that we've done for any other reconstruction. So that's sort of, ambiguous, but many other features we can actually infer a likely scenario about their evolutionary history. So for example, there are some features that are present and widespread in archaea. One example of this I already mentioned briefly, which is the type of chemical bond inside their molecules that make up the membrane. Archaea have these ether linkages, bacteria and eukarya have ester linkages. So we're going to treat membrane chemical structure as our character, and ester linkage as one character state and ether linkage as another character state, and do the same type of reconstruction. Here's one scenario. The most recent common ancestor had ether linkages, and then there was a switch to ester linkages in the lineage leading up to the common ancestor of bacteria, and a switch in the lineage leading up to the common ancestor of eukaryotes. An alternative is that the most recent common ancestor had ester linkages. There was a switch in the lineage leading up to the common ancestor of Archaea and eukaryotes to ether linkages, and then another switch in the branch leading up to the common ancestor of eukaryotes back to ester linkages. Neither of those are particularly ideal, and so this is a more parsimonious alternative, which is that the common ancestor had ester linkages, the ancestor of Archaea and eukaryotes had ester linkages, and that there was a change from ester to ether linkages in the lineage leading up to the common ancestor of Archaea. That makes sense what we're doing here? Just sort of, again, treating these as character states. So one type of character that has turned out to be really interesting and the pursuit of a lot of research, largely triggered by this rooted tree of life, has been features that have been found to be where there's a character of interest that is present in Archaea and eukaryotes and absent from bacteria. Sort of the inverse of the pattern that we had for peptidoglycan. And an example of this is histones. So you may be familiar with this in eukaryotes. So the DNA that makes up the genome of eukaryotes, like humans, can be composed of a lot of letters. So in the human genome is composed of about 3 billion letters, subdivided into 23 chromosomes. If you stretch out the DNA strands that correspond to our chromosomes, they are longer than the length of really any of our cells except maybe nerve cells. They would take up a lot of room. And what eukaryotes do to deal with this is they wrap up the DNA into coils, sort of like you're coiling rope if you're on a ship or you're a rock climber or something to that effect. And then they even wrap up these coils into coil bundles. And then those bundles into bigger bundles, and that's called chromatin. And this wrapping is done largely through the use of these proteins called histones. Histones are the proteins that serve as the center of this bundle, and you wrap the DNA around this to form this chromatin. And really nobody thought or nobody saw histones in prokaryotes before the three-domain tree of life was worked out and before this rooted tree of life was worked out. And with the rooted tree of life that we have, it shows that Archaea and eukaryotes are sister taxa. And it turns out that many Archaea have histones just like eukaryotes do. And in fact, there are many features that people have subsequently discovered about Archaea that greatly resemble features of eukaryotes that no one thought to look for because, again, Archaea do not have a nucleus, and they had been lumped together in with bacteria into this polyphyletic group called prokaryotes. And only with the rooted tree of life did people start to pursue shared features between Archaea and eukaryotes and have discovered quite a few. Many of them relate to sort of the machinery involved in DNA replication and transcription and translation. And so if we look at the evolutionary history of a feature like this, I must have inverted, oh no, red again is presence, blue is absence. So presence of histones or any of these other features that Archaea and eukaryotes share. One hypothesis is that it was present in the common ancestor of everything and subsequently lost by bacteria. And an alternative, equally parsimonious, is that it was absent in the common ancestor of everything and then gained in the lineage leading up to the common ancestor of Archaea and eukaryotes. We can't actually distinguish which of these is the most parsimonious. But just the fact of having this rooted tree of life is what led a lot of scientists to look for these features. So perhaps the most interesting features that relate to this rooted tree of life are the features that are shared by eukaryotes and different from what is present in bacteria and archaea. The hallmark example of this is the presence of the nucleus. This is the membrane compartment that surrounds the DNA in eukaryotes and is actually quite a complicated machine in essence. There are holes in the membrane that are made up of proteins that serve to channel compounds in and out of the nucleus. You have to get RNA out of the nucleus for it to be translated into protein. You have to get regulatory proteins and regulatory molecules into the nucleus in order for them to regulate transcription. So the nucleus is a really important component of eukaryotic cells. It's quite complicated in terms of its activities. And it's this hallmark feature, which is why, where the name eukaryote came from. If we want to reconstruct the evolutionary history of those features that are shared by eukaryotes, we have multiple possible scenarios again. I'm not going to go through the wording of all these, but you can just see it on the tree. The most parsimonious scenario is that these features arose in the lineage leading up to the common ancestor of eukaryotes. Now, the number one reason I'm talking about this is to get you to understand that there are many features that we will talk about that bacteria and archaea share in common. In many cases, it's the absence of something and the presence of something in eukaryotes. Bacteria and archaea look a lot alike. They have many features in common. This rooted tree of life tells us that in those cases, it is largely due to evolutionary events that happened on the branch leading up to the common ancestor of eukaryotes. And therefore, for many years, this mistaken grouping of bacteria and archaea together, because of those shared features, was a mistake. And that this model is what people think of now for the features that are shared by eukaryotes. So again, you will see when we talk about a lot of aspects of the biology of bacteria and archaea that they have a lot of things in common. That is not indicative of their sister relationship because they are not sister taxa. And the term prokaryote, which is used throughout biology, throughout the textbook, in many cases refers to bacteria and archaea. And on this tree, the term prokaryote refers to a polyphyletic group. It is not prokaryotes are not monophyletic. Does that make sense here? So the main reason I really want to emphasize this rooted tree of life is it helps us understand a lot of things. It's what led people to look for these shared features of archaea and eukaryotes. And it helps us understand that these similarities of bacteria and archaea are not necessarily telling us that they are closely related to each other. It turns out there are an enormous number of features like this. Something massive or many separate events happened in the lineage leading up to the common ancestor of eukaryotes. We will talk about some of these throughout the class. That led to a large number of differences between eukaryotes on the one hand and bacteria and archaea on the other. One of the key features that you should understand relates to processing of RNA. So in eukaryotes, DNA is transcribed into RNA in the nucleus, and then that RNA is exported to allow for translation in the cytoplasm, where the ribosomes are. This decoupling of transcription and translation is very different than what happens in bacteria and archaea, where in bacteria and archaea, DNA is transcribed into RNA, and as the RNA is growing, as you get a growing chain of the RNA getting bigger and bigger, ribosomes bind to the RNA and start to translate the RNA into proteins. It's called coupled transcription and translation. And this sort of fundamental difference in biology has set up massive differences in the way bacteria and archaea on the one hand and eukaryotes on the other go about their normal cellular functions. So for example, eukaryotes, because of this decoupling that they have, are able to do a lot of processing of the RNA before it gets translated. And so I assume you are familiar with the presence of introns in the middle of genes in eukaryotes. A single region of the genome that codes for a protein frequently codes for a lot more material than is used to make the final protein product. Regions of the RNA that are transcribed are eventually cut out in a process called splicing, where introns are removed from the RNA, and what is left are called exons, and those are what code for the proteins. This allows for all sorts of interesting regulatory processes in eukaryotes. There's something called differential splicing, where a single gene that has, say, 10 exons can be made into millions of different protein products by combining the exons in different ways. There are certain genes where one form of a protein is made from gene X in some cells, and a different form is made from the same gene by differential splicing. This does not necessarily mean that eukaryotes are more complicated or complex or higher than what occurs in bacteria and archaea. Bacteria and archaea have their own interesting features that they're able to do because of their coupling of transcription and translation. So for example, there are feedback loops where when a bacteria or an archaea is translating the RNA, it can, for example, tell when the cell is low in certain amino acids that are required to make certain proteins. That can actually cause the transcription process to directly change, and you can make more RNA for the proteins that are not being made efficiently because they're missing amino acids. Or you can make less if you don't want to waste making protein or making RNA for something when you don't have the amino acid to make the protein. So bacteria and archaea are able to recognize things that are occurring at the protein level and directly regulate transcription. Eukaryotes can't do that. Bacteria and archaea have something called operons. This is discussed a little bit in the text and is in that table that I told you about comparing bacteria and archaea and eukaryotes, where many proteins are made at once in a single RNA. A long RNA that is then translated into 1 to dozens of proteins. This is a really sort of simple, elegant regulatory process. If you want to make all of the proteins for a particular pathway or complex, you can now make one RNA and then translate all the proteins at once, and they all have the same number of RNA molecules in the cell because they're all coming from the same RNA. Eukaryotes, when they're making protein complexes, they're each coded for by a different gene that is transcribed in a different area of the genome. That has to be regulated by multiple regulatory processes across those multiple genes. So again, eukaryotes aren't necessarily better or more complicated by having the nucleus. They're different. And to understand these differences helps you understand many of the fundamental ways that bacteria and archaea differ from eukaryotes in terms of their biology. Any questions about this? Yeah?
So eukaryotes don't have those operons?
Yeah, eukaryotes generally do not have operons. There are a few kinds of eukaryotes that make large RNAs that correspond to a set of multiple proteins. They don't immediately translate those. They still have to get exported out of the nucleus for translation, but there are a few eukaryotes that have what are called, they have a different name for it, of course. They're called polycistronic messages instead of operons, but they do work in some similar ways. But the vast majority of eukaryotes do not. Yeah?
Could you just quickly explain how operons are different than
intron and extron?
Yeah, what is being shown here for eukaryotes is a single gene. So there's a region of the genome that codes for a single protein, or a single kind of protein, basically, or a single molecule of some type. It is transcribed into a large RNA. And then from that large RNA, pieces are cut out, and the ends are spliced together of the regions that are not cut out. That's why it's called splicing. And that makes the final product, which is shown here, this final message RNA, that is then translated into proteins.
Are you saying we have to copy the whole genome to get a free mRNA? Before actually
It's not the whole genome, but this whole gene, yes. And it seems to be quite a waste of energy. In fact, there are some genes in organisms that are, the final product might be 2,000 base pairs long, but the gene is 2 million base pairs long. And every time you add a single base to the RNA, that costs the cell at least one ATP molecule. So that's a heck of a lot of ATP to use to make a giant RNA that is then cut apart and sent, to the trash bin, in essence. It is used as a regulatory process, though, so it is not a completely wasteful process, but it is certainly using a lot of RNA at the start to make something that is pretty small in the end. And bacteria and archaea generally have very small genomes compared to eukaryotes. It's not clear exactly why, but one of the theories is that they are streamlined and they're selected to not waste a lot of energy, and eukaryotes seem to be able to deal with the more wasteful processes that come with large introns. So bacteria and archaea on average have, their whole genome is about 1 million to 3 million base pairs long, and some individual genes in eukaryotes are that big. Yeah. Any other? So in general, this is an example, but in general, there are a large number of processes that are sort of set up by the architecture of eukaryotes versus bacteria and archaea. The presence of the nucleus in particular sort of lends itself to many of these differences. And there are many other differences between eukaryotic cells and bacterial and archaeal cells. We'll talk about a few of those, and they include all sorts of other complexes like organelles and the Golgi apparatus and different types of flagella and different types of motility processes, but the nucleus is sort of the central organizing principle of the differences. So, partly related to the nucleus is that there are fundamental differences in how cells of bacteria and archaea duplicate themselves versus cells of eukaryotes. So in bacteria and archaea, most of them duplicate themselves by a process called binary fission. The DNA is copied, so you make a second copy of the genome, and then really the cell literally just splits down the middle, and one of the cells gets one copy of the genome, and the other cell gets the other copy of the genome. And so it's called binary fission. We'll come back to this when we talk about lateral gene transfer later, but you probably are more familiar maybe with the process of cell duplication in eukaryotes. So in normal growing times, like in the production of somatic cells in our body, or when single-celled eukaryotes make copies of themselves, they go through a process called mitosis. Many of the differences between mitosis and binary fission, you could say, relate to the presence of the nucleus. Many of the differences also relate to the fact that most bacteria and archaea have a single copy of the chromosome. And eukaryotes have 2 or more copies of each chromosome. Eukaryotes are, bacteria and archaea are called haploid. Even though they have a single copy, the term is haploid because they have, that's the term used in eukaryotes to refer to half the normal number of chromosomes. In eukaryotes, as we will talk about quite a bit when we talk about plants, animals, and fungi, there's actually an alternation of generations between haploid cells that have a single copy of the chromosome and diploid cells that have 2 copies of each chromosome. And that alternation of generation is really important in the life cycles of eukaryotes. So mitosis is the normal means by which eukaryotic cells make copies of themselves. And then meiosis is this specialized process in eukaryotes that converts cells from diploid to haploid and is used for the generation of gametes for sexual reproduction. Another fundamental difference between eukaryotes and bacteria and archaea. Bacteria and archaea do not have sexual reproduction in the same way that eukaryotes do, where in general what eukaryotes are doing is 2 haploid cells are merged together to produce a diploid organism. DNA can move, as we will talk about, between different bacterial and archaeal cells, but it is not the full merging of 2 distinct lineages in this sexual reproduction. Yeah, was there a question?
I was just wondering, how come the archaea and bacteria, they introduce to binary fission, but they have haploid cells?
How come they have haploid cells? Or
Yeah, but they have haploid cells, but how come they produce asexually?
the primary mode of reproduction of any cell really is asexual growth. So in bacteria and archaea, what they do is you make a copy of the genome and you simply split down the middle in most cases. In eukaryotes, most of the time organisms are making copies of cells asexually. Like in our bodies, all of our cells are produced by mitosis, where there's a parental cell and then it makes copies of its genome and splits in 2. The mitosis process is a little more complex than binary fission because of the presence of the nucleus and because of the multiple chromosomes that are present in eukaryotes. But it is the same concept as binary fission. You have a cell, it's going to copy itself and make 2 offspring cells. The main part that is really different in eukaryotes is they add an extra occasional process of sexual reproduction where a diploid cell is converted into haploid cells. And then those haploid cells participate in sexual reproduction and merge together from different lineages. We will talk about that more when we talk about fungi and plant and animal reproduction. Does that sort of answer your question? Okay. So again, you should, when you read the book, chapters 26 and 27, sort of give yourself guidance in there by trying to understand where some of the features that we're talking about evolved on the tree. And many of the things that are talked about in chapter 26 and 7 are not like these features that we've talked about here. Many of them, so there are very few actually that look like this, where all bacteria have one particular state, and it's different than what's in archaea and eukaryotes, or all archaea have one particular state. And largely this is because bacteria and archaea have been evolving as groups for billions of years. So there was a branch leading up to the common ancestor of bacteria. That lineage probably existed for maybe 100 million years or 50 million years early in the evolution of life. And then there's been 3 billion years for bacteria to become different from each other. Archaea may be a little more recently evolved, but not much. So there was a little bit of time for synapomorphies of archaea to evolve, but then billions of years for them to become different from each other. And so what we're going to talk about now, and what the book has a lot of examples about, and what is interesting about the diversity of bacteria, archaea, and microbial eukaryotes are really these features that have evolved on a single branch within any of these groups. And I'm going to give you at least a couple of examples of those. So one really important one that is talked about a lot in terms of bacterial diversity is a feature that has evolved, it appears to have evolved once, in the common ancestor of a group of bacteria known as gram-positive bacteria. And what this is the organization of what's called the cell envelope. The cell envelope is all of the outer parts surrounding a cell. And in bacteria and archaea, that includes membranes as well as cell walls, and sometimes other complexes, as we will see in a few examples. Gram-positive bacteria are named that way because they stain when you add a certain chemical to them that someone discovered, someone named Gram, in fact, they stain positive with this stain. And there are chemical reasons why they stain positive with the stain, but the important feature that they have is this different structure of their cell envelope. So in gram-positive bacteria, you have the inside of a cell, which is represented here in blue, and then this membrane that we referred to briefly in terms of the comparison between bacteria and archaea, and then a very thick cell wall made out of this peptidoglycan that all bacteria have. Gram-negative bacteria have the inside of the cell, and then a membrane, and then a very thin cell wall, again made out of peptidoglycan, and then another membrane on the outside of that cell wall. So they are surrounded by a double set of membranes as well as a cell wall. Now, this may seem like trivia to some people, but these differences are enormously important to the biology of these organisms. The main reason when you have, or a pet or whatever has an infection with a bacteria, and the people taking care of you, if they know what they're doing, want to know if it's a gram-positive or a gram-negative infection, is that antibiotics frequently differ in their effectiveness to these two groups. Many antibiotics target peptidoglycan in terms of how they kill cells. Peptidoglycan is both a little bit thicker in gram-positives, which makes it maybe a little bit stronger in some ways, but much less accessible because it's not, much more accessible because it's not surrounded by this extra membrane that is surrounding gram-negative bacteria. So this difference in architecture makes these two different kinds of organisms respond very differently to antibiotics and other drugs meant to target them. And it also, antibiotics are not the only thing that differs between them. The movement of molecules in and out of the cells, which is what organisms use to sense their environment around them and to communicate with each other and other organisms, is generally very different between gram-positive organisms and gram-negative organisms. So this sort of architectural difference in the cell envelope is really important in terms of understanding the biology. So for example, just the names you might be familiar with, Staphylococcus is a gram-positive. Streptococcus is a gram-positive. The causative agents of leprosy, Mycobacterium leprae, tuberculosis, Mycobacterium tuberculosis, are gram-positives. Gram-negatives include things like E. coli and a variety of Vibrio cholera, the causative agent of cholera. Bacillus anthracis, causative agent of anthrax, is a gram-positive. There are real differences in the biology of these organisms. Are there any questions about? Yeah?
You mentioned two bacteria that have become resistant to antibiotics. Does it have to do with changes in their cell wall?
Yeah, so it depends on the type of antibiotic. So not all antibiotics target the cell wall or the cell envelope. But the ones that target peptidoglycan, one of the mechanisms by which organisms become resistant is by changing some of the sort of outer structure of their cell, like how easy the chemical can get to the peptidoglycan. There are like 20 different mechanisms by which pathogens can become resistant to antibiotics. So you can export the drugs. That's a really common mechanism. That's what the multi-drug resistant tuberculosis frequently does. Just pumps. They just have pumps that are constantly pumping things out of the cell. Some of them change the surface coating so that the antibiotic, if it has to bind to something, doesn't bind correctly. So there's a huge diversity of actual mechanisms. But one of them is changing how the chemicals interact with the peptidoglycan, if that's what the target is. Any other? So this sort of summarizes some of that detail that I basically just said. So what I want to do now is sort of follow up on this gram-positive, gram-negative example and start to talk about the diversity within microbial groups. And what I'm going to do here is really talk about microbes in a very broad sense. So most of the rest of the class after the next 4 or so lectures is going to focus on fungi, plants, and animals. For the next few days, we're going to talk about everything else. And I'm just going to call them, in a sort of sloppy way, microbes. Because most of these organisms spend most of their life as single cells that you can't see without the aid of some microscope or other device. That's the formal definition of a microorganism or a microbe, is something that you need assistance to see the cells of to see the whole organism. And so we will see there are a few organisms in the groups I'm going to talk about that are actually multicellular. Kelp, not a microbe, it's enormous, right? But we're going to basically talk about the rest of diversity outside of fungi, plants, and animals, most of which is microbial. So microbes have been used and manipulated and indirectly studied for a really long time. Anybody doing fermentation was making use of microbes. We've been doing that for thousands and thousands of years. Bread and all sorts of other food products, we've been making use of microbes. But people really didn't understand very much about them until the invention of the microscope, which was in the 1600s. And then some people turned microscopes towards looking at things like the human body and human cells. That's where the first actual cells were observed. But this one person in particular, Antony van Leeuwenhoek, started taking a microscope. This is actually a picture of one of the microscopes that he used. It's really simple. To look at the world around them and look at microorganisms. And, for a little while, maybe they weren't particularly appreciated, but over the years, long before any of the sort of molecular stuff that we've been talking about, over the years, people have begun to appreciate the enormous diversity that you can see in the microbial world. And not just the diversity, but the importance. So in a gram of soil, which is sort of about maybe a little bit smaller than this piece of chalk, there are at least 100 million cells of microbes in most grams of soil. Massive numbers on the planet. More cells of microbes on Earth than there are stars in the universe. They're small, so that's not really that big of an accomplishment, I guess, but their total biomass is greater than the total biomass in plants and animals and other, fungi, the multicellular creatures. The thing that both is really interesting and that freaks some people out is that in and on your body, there are 10 times as many microbial cells as there are human cells. Thousands of different species of microbes swimming and crawling all over you. We will talk about these more later in the class. But, microbes basically contribute or run the biology of the planet in many ways. And when you look at the massive diversity of microbes, what we're going to do is sort of walk our way through some different aspects of this diversity. So we're going to look at the diversity of form, that is sort of the appearance of microbes. That's really all people knew for a while when they were first taking microscopes and looking at them. Before anybody had even taken a microbe and isolated it in the laboratory, you could really just look at them. You can study how they move when you look at them in the microscope, and we'll talk a little bit about different ways that microbes move because it's just really cool. And then we'll spend a little bit of time talking about the diversity of functions of microbes and the diversity of kinds, that is, the phylogeny of microbes. So here's sort of a summary slide with a lot of different examples of the diversity of form in microorganisms. There are, for example, corkscrew-shaped or spiral-shaped bacteria. There are incredibly complicated single-celled eukaryotes with extracellular structures that are both gorgeous and very complex. They form excellent fossils. There's a great fossil record of many of these. There's multicellular bacteria, like this and this, that actually form large structures. There are bacteria like this one that is about the size of the head of a bee. So there are some bacteria you can see without the aid of a microscope. There are, multicellular eukaryotes that are not fungi and plants and animals we will talk about a little bit. This is one of my favorites because we actually work on it a tiny bit in my lab. This is a single-celled eukaryote isolated from the bottom of the ocean where I do a little bit of research, picked up by the Alvin submarine. It's the size of your fist. It leaves trails in the mud, and it's a single cell. So there's a lot of diversity out there in terms of what we might, what I'm lumping together into calling microbes. That is, again, everything except plants and animals and fungi. And this diversity is completely overwhelming in many ways. And what, everybody does when we're confronted with a lot of diversity is we try and classify it into a variety of ways. So one way that people classify shapes is these three categories, which are talked about a little bit in the book. So in bacteria and archaea, most of them, though not all, can be classified into these three different forms based upon the shape of the cells. Anything that's called coccus is generally called that because the cells are sphere-shaped, a ball. They might form collections of balls together, but Staphylococcus, Streptococcus, Lactococcus, all of those, the cells generally form this sphere-like shape. Other cells have a rod, an elongated, like a piece of chalk, basically, an elongated rod-like shape. That shape is called a bacillus. So many of the organisms, Bacillus anthracis, for example, the cells are rod-shaped, and you'll hear people refer to a bacilli. That's generally referring to the rod shape of the organisms. And as I showed you this picture of, there are some bacteria and archaea that have this spiral corkscrew shape. We can do the same thing with eukaryotes. Really, these are, I wouldn't call these shape, but these are general classes talked about in the book in terms of the body types, you could say. So ciliated cells are cells that are surrounded by these little hair-like projections called cilia. They're used for movement and carrying out other types of processes. We'll see this in a little bit. Flagellated cells have a flagellum, which is a long whip-like tail. They might have many of them. You also see flagella in bacteria and archaea. And then everything else is frequently dumped into the amoeboid shape category. That is the blob, like white blood cells that you might be familiar with, or as we might get to talk about, the brain-eating amoeba that's now in the press again because there's a massive outbreak of this somewhere in Pakistan right now. So this is a picture of one of the multicellular bacteria. It's in a particular group of bacteria we'll talk about later called the Proteobacteria, but it actually forms these fruiting bodies, and there appears to be division of labor between the different kinds of cells, and it has structures much like, in small form, much like maybe a mushroom. There are bacteria that gang up together, this is talked about a little bit in the book, to create what's called a biofilm. It's in essence a thick layer of cells. And biofilms are in particular important in a lot of environments because they form these sort of stable structures of a particular kind of microbe. They're also really important in medicine because when bacteria form a biofilm, they tend to be very resistant to antibiotics because it's hard to get antibiotics into the middle of the biofilm. You can sort of kill things on the outside. Biofilms form, for example, on a lot of implants, on artificial organs and things like that, as well as in various parts of the body, in your veins and in blood vessels, and they can be really recalcitrant to treatment. So bacteria don't spend their life as just single cells floating around, looking for a neighbor and a buddy all the time. A lot of microbes form structures. They might be, uniform, that is all the same kind of microbe together in a clump. They might be diverse in terms of being sort of like a multicellular organism. What I'm just going to end with here is some discussion of motility of the rest of the tree of life. There's some really cool ways that organisms move around, and probably the most important thing to realize here is that it's not just, animals that are able to move themselves around in the world. Even single-celled bacteria have the ability to move, to sense their environment, to move towards things that they want or away from things that they don't want. And they're able to spread around in a variety of ways. There are a lot of different mechanisms by which organisms are motile. Some of them use this, a flagellum, which is this long filament that rotates. The bacteria have flagella, Archaea have flagella, Eukaryotes have flagella. They appear to all be distinct, non-homologous. All the bacterial flagella appear to be homologous, and then it's different from what we see in Archaea and what we see in eukaryotes. Many eukaryotes have cilia. The best example of this that you might be familiar with is the paramecium. It's in a group called the ciliates, as we will hear about in the next lecture. There are also organisms that glide. There are others that tumble and roll. And a lot of them, especially aquatic ones, a lot in water, control the depth that they're at by using gas bubbles, sort of like submarines can change their buoyancy by changing the amount of air or water they have inside these bubbles. So here's an example of flagella in a bacterium. I think this is Salmonella, although I'm not 100% sure. It's got quite a few flagella that it uses to move around. The flagella is actually a very interesting, very complex structure. The anchor of the flagellum is buried into the cell membrane, and there's some really interesting structural biology on the details of the flagellum. This is a video of some of these, I think, Vibrio cholera. Seems kind of pointless for this one here, but the other ones seem to be going somewhere, maybe. This is a Dinoflagellate, a eukaryotic microbe we will talk about in one of the next lectures, pulsing with its cilia to move water, and that's how it feeds itself. Yeah, I would